आत्मोदकं सदा विमुक्तिसाधकं कलाधरावतं सकं विलासिलोकरक्षकम्.
Studied about the different, you have listed the cell organelles there. Yes, you have studied about the cell membrane, cell wall, nucleus, cytoplasm, all these things we have studied. Now we will go one by one the different cell organelles which are present in the cell there. The first one that we are going to discuss is about endoplasmic reticulum. In short, we call it as ER. In short, we call it as ER, endoplasmic reticulum. So the structure of the ER, that is endoplasmic reticulum, is similar to that of the plasma membrane. So if you observe the structure of your endoplasmic reticulum, it is somewhat similar to the structure of your plasma membrane there. It is a network-like structure which consists of membrane-bound tubes and sheets. So you can see here the endoplasmic reticulum. This is your nucleus. So you do find this endoplasmic reticulum the very next to your nucleus or the nuclear membrane there. This is your nucleus. This is your nuclear membrane. The very next to whatever you find, yes, this is your ER. That is endoplasmic reticulum. So it is a network-like. So if you observe, it is just like a network here. It is network-like structure which consists of membrane-bound tubes. So you do have the tubes type also, or in the form of sheets also. You do have the tube structures also. With not just that, we do have the sheets also. In case of your endoplasmic reticulum, there are majorly two types there. We do have two types. Those are rough endoplasmic reticulum, smooth endoplasmic reticulum. We call them, the two types are rough endoplasmic reticulum, yes, and the smooth endoplasmic reticulum there. So yes, now let's go through one by one what function does a rough endoplasmic reticulum will be doing, what function your smooth endoplasmic reticulum will be doing there. So moving on to the rough endoplasmic reticulum. So we call it as a rough endoplasmic reticulum. The reason is because it contains ribosomes that are responsible for the manufacturing of proteins in the cell. So we call this rough endoplasmic reticulum. That means the first type, that is rough endoplasmic reticulum, because they do have, yes, they do have what? The ribosomes. You are able to see? Yes. So these are what? Ribosomes there. So they are situated inside your endoplasmic membrane there. That's why we call it as rough endoplasmic reticulum, because they have the ribosomes. So these ribosomes, yes, they play a very important role. That means they are responsible for the manufacturing of proteins in the cell. So whatever the proteins which are required by our body, those are being synthesized inside our body by these ribosomes there. They give a rough texture to the cell. So as these ribosomes are present on the surface of your endoplasmic reticulum, they give a rough surface structure. They are not having smooth surface structure here. Okay. Now the smooth ER, that is smooth endoplasmic reticulum. The smooth endoplasmic reticulum manufactures fats or lipids. So this is very clear now that rough endoplasmic reticulum, it is going to help, or as it is having the ribosomes with it, they are mainly having the work of production of proteins there. Now when we are talking about the smooth endoplasmic reticulum, they manufacture fats or lipids in the cell which allows the functioning of the cell. So this fats or lipid, as we have studied in the plasma membrane, we do have the lipids as well as the protein constituents in the membrane. So these fats and lipids are very, very important there. what are the functions of lipids and proteins? So as we are seeing that there is a function of proteins, that means production of proteins there. At the same time we have the production of lipids, fats. Why are they important? What is the reason there? So proteins and lipids synthesized on ER are used for making cell membrane. So whatever the proteins or your lipids or fats which are being synthesized on the ER, they are again sent to the boundary line. So if this is your cell, this is your nucleus, so you have your endoplasmic reticulum here, correct? So whatever the things are synthesized, they will be sent to your cell membrane. So they are used for making the cell membrane there. They are used for making the cell membrane. The process is known as membrane biogenesis. So this particular process is called as membrane biogenesis. It is called as membrane biogenesis. Proteins can act as an enzyme. So proteins, they have the ability to act as an enzyme. It means the messenger molecules. Okay, both proteins and lipids can act as hormones also. So they have the ability, yes, to act as hormones. It means these are chemical substances which are very, very important in a body. Each part in a body do have these enzymes. We do have, in the stomach we have digestive enzymes. In liver also we do have some of the enzymes. Yes, these enzymes do play very important role here. So these proteins and lipids, they may act as the enzymes as well as the hormones there. They can act as the hormones. Now, moving on, what are the major functions of your endoplasmic reticulum? As we have seen the functions of your, that means what they are, why are these important as your rough endoplasmic reticulum or it might be smooth endoplasmic reticulum. Now, on whole, what is the major function of this endoplasmic reticulum there? Transportation of materials between different parts of the cytoplasm and also between the nucleus and cytoplasm. So, the major function of your endoplasmic reticulum is transportation. Yes, the transportation of materials between different parts of the cytoplasm. So, if in your cell you have all this cytoplasm, this is your nucleus. So, different materials throughout your cytoplasm, they have to be moved. So, this endoplasmic reticulum plays an important role there. Is it only of that? No. And also between the nucleus and the cytoplasm. So, it acts as a transporting agent not just throughout the cytoplasm but also your nucleus and the cytoplasm there. Folding of proteins which are synthesized by ribosomes on RER. So, it is in depth that when the protein synthesis takes place, okay, they will be in the chain form. Yes, so they have to be folded in a proper, whichever protein is required by a body. Yes, it is being synthesized, but in a usable form, if we want it, then it has to undergo what? Folding. That means folding of proteins takes place. So that is in RER, ribosomes. They are what? Synthesizing these proteins. So this ER is involved in folding of your proteins there. Then detoxifying poisons and drugs out of the cell is the function of SER. So RER is involved in the protein synthesis, at the same time it is also involved in your protein folding. Now when we are talking about SER, smooth endoplasmic reticulum, the first function is it helps in synthesis of lipids and fats and all which are required, which are being taken to the synthesis of new cell membrane there. But with that, they help in detoxifying poisons, whatever the poisons is being synthesized. Yes, we can take the waste also as the poisons at some time. So they have to be removed from the cell. So it is the function of your smooth endoplasmic reticulum. It is the function of smooth endoplasmic reticulum. Now moving on, the next cell organelle that we are going to study is the Golgi apparatus. It is also called as Golgi apparatus or Golgi bodies. So Golgi apparatus, Camillo Golgi. So who is the founder? That means who found these Golgi bodies in the cell? It was Camillo Golgi discovered the Golgi apparatus. We call it as Golgi apparatus or Golgi apparatus. It contains vesicles that are arranged parallel in stacks. These stacks are called cisternae. So you are able to see this, yes. So they are arranged in a parallel, yes, they are parallel to one another. Yes, they are parallel to one another. At the same time, yes, they are stacked. They are like in stacks. So these are called as cisternae. They are called as cisternae. They are called as cisternae there. So these vesicles have their own membrane. So these all vesicles, whatever you are able to see, they have their own membrane. They have their own membrane. So these membranes are sometimes connected to those of your ER. That means, so these membranes, whatever you are able to see here, they are sometimes connected to the endoplasmic reticulum also. They are also being connected to the endoplasmic reticulum. So you can see, yes, we have, this is the structure, stack-like structure. So we have vesicles, we'll come to know in the next, vesicles, yes. So we have lumen, we call it as cis-phase. So what all does the function here? Now we came to know this is so and so structure. So it is like this and it somewhere it is helping in the transportation. But what are the major functions do we have here of the Golgi apparatus? Golgi apparatus carries materials synthesized by ER to different parts of the cell. So this is very important. It is like a transport vehicle. What all things, materials that are being synthesized in your endoplasmic reticulum, they are being carried to the different parts of the cell by this Golgi apparatus. So if you have seen here in the structure itself, yes, transport vesicle, we are having them as transport vesicle. So if anything comes inside, it is going to move out in this vesicle form. That means the messenger, it is being covered with this, yes, it comes inside, slowly it forms the loop, yes, and we have the vesicle formation and it is moving. So in this way the transportation takes place within the different parts of the cell. That is the main function here. This material is sorted and packaged in vesicles. You just saw that we are being whatever materials that you want to transport, they are being packaged in the vesicles there. Formation of complex sugars. Yes, formation of complex sugars. So the major complex sugars that you can take here is, you have your starch, or you can take the cellulose, you have the cellulose there, yes. Formation of lysosomes. So lysosomes are also a type of organelles which are present in the cell. Yes, we'll study in the next classes there what are the functions of lysosomes. So these lysosomes, they are like, yes, suicide bags. We call them as suicide bags because they are going to take all the dead and decaying things which are being given out in the cell, yes, and they are not just taking it, they are going to completely remove them from the cell. They are completely destroy all those wastes from the cell there. Yes. The very next that we have is lysosomes. We just saw that the Golgi apparatus is very much important in the synthesis of your lysosomes. So let us study about the lysosomes here. Okay, so the lysosomes, they are single membraned vesicles. This is very important. They are single membraned vesicle. They don't have double membrane as in case of your cell membrane. They are what? Single membrane vesicles which are responsible for clearing the cell. That means all the waste, whatever you have, this is the membrane. Yes, so this lysosome, it has different kind of proteins or enzymes in it. It contains different kinds of proteins or enzymes. Okay, so it is very much important because it is going to remove all the waste which are being produced inside the cell. They have been completely removed from the cell. Yes, that is the main function of your lysosomes here. So they can digest any foreign material such as food, bacteria, and even the worn out cell organelles. So this lysosome is very important because it is going to digest, that means completely remove the foreign materials, whatever the foreign materials that we are getting or which are entering in the body. They might be the food. Food is also your foreign material only. It is not inside our body. We are eating it from outside. At the same time, we have your bacteria, the microorganisms may be involved, and even the worn out cell organelles. Sometimes what happens, if the cell gets damaged there, you get tears on the organelles. So if that particular thing is occurring there, then these lysosomes, what they do is they just engulf that particular organelles, and they are going to use, because these lysosomes, they are having the special kind of proteins or the enzymes in them. So these proteins and enzymes have the ability to completely destroy. That means if it is a complex substance, these proteins are going to attack on this and they form very small substances. Very small substances. Complex one to a simpler product, and sometimes what happens, these simpler products are again some of them, not all of them. Some of them are again being used by the cell, and the completely unwanted one are given out of the cell there. Yes, how lysosomes can digest any foreign material that enters the cell? That's the big question, huh? Yes, how are they going to do? So lysosomes are capable of doing so because they have digestive enzymes in them. Yes, they do have the digestive enzymes. So these enzymes break the materials and digest them. So these enzymes are synthesized by RER and packaged into lysosomes by Golgi bodies. Yeah, this is very important. Now we just came to know that they do have different kind of enzymes, they have proteins. Yes, so the question comes, from where does these proteins or enzymes are going to be there in case of your lysosomes? So these proteins or enzymes, they are being synthesized by the RER, that is nothing but rough endoplasmic reticulum. We have already known proteins or the enzymes, they are being synthesized by your rough endoplasmic reticulum. So rough endoplasmic reticulum, it synthesizes it, then it is being transferred to your your RER. They are again transferring it to your Golgi bodies. From there they are being packed into vesicles, and then they form your single membrane lysosome there. Why lysosomes are called as suicide bags? I said they are called as suicide bags. What is the reason there? If the cell's own material get damaged, if the cell's own materials get damaged or dead, either they get damaged or they are completely dead, there are chances that lysosomes burst out, thus digesting its own cell. So if you take if this is your cell, okay, so now you have the nucleus, you have all other cell organelles in it, and this is your lysosome. I'll take different color for that. Yes, so this is your lysosome there. So what happens if the cell organelles of in your cell itself, if you have tears or any kind of breaks there, so this lysosome, it completely gives out all its digestive enzymes outside, and yes, the complete cell is being digested. It completely engulfs, andre it takes complete of its cell. So that's why it is called as suicide bags. They are called as suicide bags there. Okay, the next very important cell organelle is the mitochondria. It is the mitochondria. So it is a double membrane organelle. So now please be careful here. Mitochondria is double membrane organelle. So till now we have not read anywhere it is double membrane. In lysosome it is very specifically mentioned it is single membrane. Here mitochondria is double membrane structure which has its own DNA, and that is why often called semi-autonomous organelle. So as we have studied in the nucleus contains the genetic material, that is DNA. No, not only the nucleus has the genetic material. Here mitochondria is one such organelle which has its own DNA. That means it has the ability to multiply itself. DNA helps in multiplying the different cells there. At the same way, your mitochondria, it has its own DNA, so that it can, if it wants to increase its number, it can increase by itself. It did not require anyone's orders there. It does it by itself. The cell requires energy in order to carry out several activities. If the cell has to do a lot of work, it requires energy there. This energy is generated by the mitochondria, which are often called as the powerhouse of the cell. So the mitochondria, it is called as the powerhouse of the cell. Mitochondria are site of cellular respiration. So mitochondria is the site for cellular level respiration. That means cell level respiration. They use oxygen from air to oxidize the carbohydrates and thereby release the energy. So what is the main simple way is they take up the oxygen from the air, whatever the food, that means carbohydrates, proteins, whatever they are present in a food, okay, when we are doing digestion process. So what happens, they are simple form to carbohydrates, proteins and lipids. So they are being taken to the cell. So here mitochondria takes those carbohydrates, then it forms simple forms. Okay, what are energy currencies of a cell? So what are different energy currencies of a cell here? So the mitochondria generates ATP, that is adenosine triphosphate. We call it as adenosine triphosphate, which are energy giving molecules of the cell that are often called as energy currencies. So these ATP molecules, that is adenosine triphosphate, ATP molecules, they are called as energy currencies because our cell is able to take the energy only in this form, not in any other form there. The two membranes of mitochondria. Yes, now here, as we said, they are double membrane structures. They have two membranes, the outer membrane and the inner membrane there. Outer membrane, it is porous in nature. The outer membrane is porous in nature, whereas the inner membrane is it is completely deeply folded there. The inner membrane of mitochondria called as cristae. So these foldings, yes, they are called as what? Cristae. They are called as cristae. Facilitates generation of ATP molecules as it has large surface area. So the reason why the inner membrane is folded is it facilitates generation of high amount of or higher number of ATP molecules there. Okay. Yes, all right. So then comes, what would be the function? We have already, yes. So the main function of your mitochondria, is energy. It is energy production. Okay. Now today we have studied about endoplasmic reticulum, Golgi apparatus, lysosomes and mitochondria. In the next class we will study about plastids, then we have vacuoles and other cell organelles. Yes, thank you.